Hello, this is Masaki. Now it's the time for researchers who can. We introduce the researchers from all over the universe. Today's researcher is FREUDIGER from Harvard University. Hello.
Hey, how are you?
Nice to meet you. First, I want to, could you tell me about your research thing?
So I work on light microscopy, especially I work on a technique called CARS microscopy, which is an abbreviation for Coherent Anti-stokes Raman scattering microscopy.
How the CARS works?
So in CARS, instead of just having one laser illuminating the sample, we have two lasers. And the two laser colors, they mix and produce a third color. And depending on the intensity of the third color, we can say whether the one chemical species is present in the sample. So basically, in a CARS microscope, it works like a TV, where the beams are scanned through the sample. In a TV, the beams are scanned through the screen to produce the image. So we scan these beams, and for every pixel in the sample, we do this measurement, and therefore we can build up this map of the distribution of a certain chemical.
Mm-hmm. What is the advantage of CARS?
So the advantage of CARS is that it's a non-invasive technique. It does not require to have any kind of label in the sample. So we can take, for example, the pure skin and look at it and look how chemicals are distributed in the skin. The grand advantage is that we can do it in live patients, for example, because we do not have introduced any extrinsic labels.
Mm-hmm. Let's do the next topic. What can you offer to other researchers?
So currently we have 3 or 4 microscopes being set up which are completely functional, and these microscopes are expensive. So if people are really interested in saying like, well, we have a great sample to look at, we are always very happy to have visitors come and try the samples, and if it works out, we can probably like go on and have a continuation of the project. As a second thing, actually, we offer annual workshops where we invite like 40 to 50 people every year and teach them how to do CARS microscopy. And the great thing is for students, we can normally offer scholarships which can bring the students even to America.
Mm-hmm. That's great. Mm-hmm.
Yeah.
Let's do the third topic. Could you tell me your future research then?
So currently, basically, we build up these microscopes and they're working. So in the next year or so, I want to apply CARS microscopy to imaging certain disease of the central neuron system. So especially like the disease in the brain. And we want to focus on tumors, we want to focus on demyelination disease, we want to focus on stroke.
Why do you want to use CARS?
So currently, if we think about a scenario that the patient is diagnosed with brain cancer, in a surgery they would take off the skull, take either a knife or scalpel and cut the tumor out or suck it out. But basically, while the neurosurgeon does the surgery, he does not know what he cuts, whether that's like still the tumor or whether it's already healthy tissue. And of course, you don't want to cut out any kind of healthy tumor, because then the poor guy might forget the name of his wife, for example. So you really
it's a big problem.
Yeah, exactly. So you really want to know where the tumor is and where the healthy tissue is. So far, they would cut it, and then like it goes to like diagnosis, which typically takes like half an hour to an hour. And that time the patient sits there with his open skull during the surgery and has to wait until the diagnosis, and then actually the surgeon can go and do the next step or close the skull. So with CARS we could do that in vivo. We could go in there because we do not have any extrinsic labels. We go into this tissue and can look at it. That saves a lot of time, and in the end will decrease mortality.
Could you tell me your motivation to the research?
So I'm really motivated by the fact that it can actually help people. It's not research for the sake of research, like a lot of physics experiments maybe are these days. But we do the research, and in the end we decrease mortality during these kind of surgeries.
Well, why did you get interested in CARS?
So what's great about CARS is that it's a very interdisciplinary field. So I'm a physicist, but I work with people in the medical field. I work with biologists. So it's something in between the disciplines, and I'm really motivated in doing this kind of work.
Let's do the next topic. What you need to achieve your research?
So that kind of ties in with the last point that I said we are working in a very interdisciplinary field. That also means I don't know everything I'm working on. So my background is in like knowing how to build these microscopes and how to operate them. But I really need help with the biology or the medical questions. So in the example I gave you before on the brain, I needed the help from people providing me with the animal models. So like I'm not sure how to build up like a stroke model in mice, for example. So I have to learn that from other people, and really I need this help to be able to like do my research. On the other hand, if we try to diagnose the disease, we compare it to existing technology, which is histology. So I need people who are very like aware of how to use histology to diagnose these kind of disease. So I need people to help me with the interdisciplinary research.
Okay, it's time to finish. Thank you. See you next time.
See ya.
See you.